Prachi Shukla. I'm a master's student at ETH Zurich, and I'm going to talk to you about a score that I created to differentiate between epithelial and mesenchymal cells in cancer and normal tissue. So, why do we need to do this? Well, basically because of a process called epithelial to mesenchymal transitions that occurs normally in embryonic cells in embryogenesis and is also dysregulated in cancer. And so basically what happens is we have epithelial cells, which as you all know are in organs all around our body, and they're normally stuck to basement membranes. They're tightly bound together with junctions. But in cancer, it's dysregulated, so a lot of these junctions disappear, and they start to move away from their basement membranes. And they become motile, and as a result can move around the body and can seed tumors in various portions, so metastasis. And as they transform into mesenchymal cells, they go through different changes. And these hybrid phenotypes that are the middle three here basically have a lot of multipotency, so that it can become different cells, and that stemness can make them more likely to seed tumors. And so it's quite an interesting thing for treatments, for cancer treatments, and for preventing metastasis. So, as I said, they're epithelial to mesenchymal transitions are associated with metastasis. They're also associated with drug resistance and poor patient survival, obviously. So what we want to do is we wanted to create a score that could characterize epithelial, mesenchymal, and hybrid cells. And the purpose of doing this is that it would be helpful in cancer research to be able to identify cells that are in between these two phenotypes so we can study them better. It'll be useful in cancer treatments if you get cells, circulating tumor cells that might be from hybrid phenotypes, so you can classify whether either a patient is responding to treatment based on how hybrid the cell is, also whether or not they have any hybrid cells and what that means for the patient. So why the score is better than the current method? Some of the current methods that are used involves just looking at marker expression, so Western blots, for example. And this isn't very good because marker expression can vary significantly across patients. It can vary across tissues and different cancer types, so it doesn't work great. You can't really use them to compare across studies. Histology can be very subjective, obviously, even with 2 or more people making assessments. And so our score can be used across studies. It can be used across tissue types, which is great. Okay. So basically what we did. So we looked at TCGA data and GTEx data. We combined the transcriptomic data, and we labeled the samples as either epithelial, EM, or so hybrid, or mesenchymal. The EM samples were left out of our training data. It was mostly a designation to separate samples that we would use in later analysis. We learned gene weights using linear discriminant analysis, and then we tested the score on some external data sets. So these are the results we got. This is after we got the gene weights. We applied it to TCGA, which is the image on the left-hand side, and GTEx, which is the image on the right-hand side. And basically what I want you to take from this picture is, 1, our score does separate epithelial and mesenchymal tissues. 2, it's beneficial to look at these tissues separately, obviously, because there's a huge distribution of scores, and it's beneficial to look at them differently. Each point here is a sample. I should mention. So the way the score works is as it increases, the tissue becomes more mesenchymal. So we looked at all our samples and we plotted them. Here again, TCJ on the left and GTEx on the right. We plotted them based from decreasing to increasing score, and we plotted them against epithelial and mesenchymal markers, which are the 2 plots below, epithelial on top and mesenchymal below. And as you can see, as the score becomes more mesenchymal when moving from left to right, you see a decrease in epithelial marker expression and an increase in mesenchymal marker expression, which is really good. So we did another test to verify that this works. We looked at uterine cancers. There's 3 different types of uterine cancers. There's the epithelial carcinoma, there's the mesenchymal sarcoma, and then there's the hybrid, which is the uterine carcinosarcoma. First, we verified their phenotypes using some marker expression, just to give you an idea of what it looks like. We verified in other ways as well, but basically, as you can see, the carcinoma has high epithelial marker expression, the sarcoma has high mesenchymal marker expression, and the hybrid is sort of in the middle. And our score reflects this. So on the right-hand side, you can see that again, reminding you that our score as it increases, it becomes more mesenchymal. So you can see that the carcinoma score is quite low, the sarcoma score is quite high, and the hybrid sample score somewhere in the middle. So I tested this also on some external data sets. I'm only going to show you one because I don't have enough time. So the data set used here, it's in cell line data. Basically, the researchers took two non-small cell lung cancer cell lines, and they treated them with TGF-beta, which induces EMT. And so this is pre-treatment and after-treatment samples for these two cell lines. And on the left-hand side, you can see marker expression that shows that EMT did occur because mesenchymal marker expression, which is CD44 and vimentin, increases while epithelial marker expression, which is eccohesion, decreases. And you can see the same for both cell lines. The top one is H358 and the bottom is A549. And our score also reflects this. You can see that the mesenchymal character of the tissue does increase when TGF-beta is applied, and our score scores it as mesenchymal. Another study that the same paper did is a time course where they looked at a time course. So the samples at different time points. And so what we did here basically is we plotted common common transcription factors or epithelial and mesenchymal markers to track to see if our score does correlate with their expression. So if you look notably at point 72, at time point 72, there's a small blip. And if you look at the marker expression, where here in the two bottom graphs, the top graph is epithelial marker expression, the bottom graph is mesenchymal marker expression. And you can see that there's a small decrease in epithelial marker expression, a small increase in mesenchymal marker expression, which relates with the small increase in mesenchymal marker expression that our score shows. So that does hint that our score does actually show us an EMT phenotype occurring. And I should mention, another benefit of using TCGA and GTEx data is that we can get reference samples, which we use for every cancer type, which is nice to just get an idea of what our samples initially look like compared to a large portion of data that we get from these databases. Okay. So why the score is good? Well, one, it uses references from a whole number of tissue types from normal and cancer tissues. We look at a lot of cancer tissues, which is really helpful because most scores don't, because it's hard to. It's really simple to use. You only need counts from transcriptomic RNA-seq data. The fact that it's trained on a large amount of data is great because there's a lot of variation in mesenchymal marker expression, and it can be used on many different data types. I know I didn't show you that, but we have done it. So there are obviously some issues. One is the classifier imbalance. We did have a lot more epithelial samples than mesenchymal samples, and that's sort of just the nature of the way things are. There's a lot more carcinomas, and they're studied a lot better. Most classifiers have some sort of imbalance, but one of the main issues that we actually did run into is tissue quality. So GTEx tissues are actually a mixture of epithelial and mesenchymal cells. So maybe not it could possibly muddy the data slightly. We didn't really see that when we ran many tests on external data sets and some of the other case studies that we did, but obviously we need to test that a little bit more. There were also TCGA adjacent samples that we used in the training data, which we're not entirely sure where they come from since they're collected by different people. But they made up such a small portion of our data set that it shouldn't really affect the training data. And these are my references. Thank you.
Did I talk too fast? No. Okay. Are there any questions?
Yeah. Yes.
But then what do you base your label on? Is it
Okay, so the question is, how do I label cell sample?
you said on the slide before that basically how people are labeling it right now is subjective. But then if you use the same for training, then you can't train on you're training on something that you say is already subjective.
Yeah, so we didn't actually just train on the marker expression. We also looked at the clustering of the samples based on our very large gene set. Oh, so the question is, how do I label samples, epithelial, hybrid, or mesenchymal? We did look at TSNE, PCA, and we did also look at marker expression, but we included a number of different markers, and we just didn't use the two most common ones, which I showed you in the external data set. So we did do it in a couple of different ways. Yeah, so obviously the ones that we couldn't definitively designate, we left out of the analysis. But there's also literature that suggests certain tissues might be, a certain phenotype based partly on knowledge of embryogenesis, partly on other studies, including gene expression. So we used all of that together to label these tissues. No?
If there are no more questions, we will continue with the flash talks.